DNA helicases are essential enzymes that unwind the DNA double helix, which makes it accessible for replication and repair. Helicase is made of 6 subunits that are arranged in a ring around single-stranded DNA. Helicase binds to the lagging strand template and unwinds the helix in a process that depends on adenosine triphosphate, or ATP, therefore it consumes energy to separate the strands. Let's take a closer look at how DNA helicase functions during replication. During the initiation of DNA replication in bacteria, the helicase DNA B must be loaded onto single-stranded DNA, ssDNA, to begin unwinding the double helix. However, DNA B forms a closed ring structure and cannot load onto DNA by itself. This process requires the assistance of the helicase loader, consisting of proteins DNA C assembled into a hexameric structure. DNA C binds to DNA B in the presence of ATP, inducing a conformational change that opens the DNA B ring. This DNA B-DNA C complex can then encircle the ssDNA at the replication origin. Once properly positioned, ATP hydrolysis triggers the release of DNA C, allowing DNA B to close into a hexameric spiral around the ssDNA. This spiral is analogous to a lock washer. Each of its 6 subunits binds 2 nucleotides non-covalently and is linked to adjacent subunits by domain-swapping sequences. DTTP, which binds to the subunits of the DNA B helicase, drives conformational changes that result in sequential shifts of the subunits in a hand-over-hand motion. This movement advances the helicase by 2 nucleotides per step in the 5' to 3' direction, which pulls the DNA strand through and unwinds the double helix efficiently. For example, subunit F moves to the position previously occupied by subunit A, while A advances further along the strand. Following this hand-over-hand mechanism, subunit E then shifts to the former position of subunit F, and subunit D takes the place of E. This coordinated sequence continues with each subunit cyclically stepping forward, pulling the DNA strand through the central channel of the helicase. In conclusion, DNA helicases are essential enzymes that unwind the DNA double helix to make it accessible for replication and repair. They are made of 6 subunits that form a ring around single-stranded DNA. DNA C plays a critical role in loading the DNA B helicase onto single-stranded DNA by opening the DNA B ring in an ATP-dependent manner, enabling proper helicase assembly. Using energy from ATP and DTTP, helicases move along the DNA in a hand-over-hand motion, advancing 2 nucleotides at a time. This energy-driven process separates the DNA strands, allowing the replication machinery to copy the genetic material efficiently.